Hey everyone, welcome back for Chapter 6. Hopefully Exam 2 went well, and you will be relieved to know that the next exam only has 2 chapters on it. They are a little more in-depth and there's more detail that has to be memorized for them, but overall it is less material. So let's go ahead and get started. So Chapter 6, we get into the beginnings of metabolism, and we have to talk about energy a little bit, kind of lay the groundwork. So energy is the ability to do work or to bring about change. And ultimately, all life on Earth is dependent on solar energy. Depending on how you want to argue it, there are some potential exceptions where we've drilled down a few miles into the ground and found rocks that are living off of not rocks, but black mold that are living off the radiation from the surrounding rocks. Kind of how plants use solar radiation for photosynthesis, but you could argue that's still dependent on the sun because the Earth wouldn't exist without it. But that aside, photosynthesis provides the nutrients for just about everything on the planet, aside from a few very specialized chemosynthetic organisms that live around like sulfur vents and things. So, forms of energy. We have kinetic energy, which is, something's physically moving. Then we have potential energy, and that potential energy is stored. And you can either store like gravitational energy or chemical energy, like stored in molecular bonds, which is basically what food is. And refer to the differentiating here, we have chemical energy, which is stored in chemical bonds, and mechanical energy is the energy of motion. So I guess you could kind of say that gravitational potential energy is stored mechanical energy. Which takes us now to thermodynamics. And again, this is just a real brief overview. This isn't a physics course. You just kind of have to know some of the principles in order to understand where we're going later. So, first law of thermodynamics, energy cannot be created or destroyed. You can only convert from one form to another, and this is referred to as the law of conservation of energy. Technically, you can also convert mass and energy back and forth, and there are a fair number of arguments for matter actually being a form of highly organized energy, hence the ability to convert them. But we're not going to get into that because that's graduate level physics right there. But for our example for this, we have the solar energy from the sun being converted into chemical energy inside the chloroplast of the plant, and a fair amount of this is lost as heat, but none of it is destroyed. Second law is energy cannot be changed from one form to another without the loss of usable energy. Essentially, there is no process that is 100% efficient. This is why, or one of the big reasons why, perpetual motion machines do not work. And if you could find a way to have something move perpetually, it would essentially be a heat engine that would eventually immolate the rest of the universe. The second law essentially states that nothing can be moved with 100% efficiency. So according to these two laws together, no process requiring energy conversion is ever 100% efficient. And for our definition here, heat energy is not useful because it is not available to do work with. You have to have a ton of heat concentrated in a very specific area for it to be useful for anything, and really that's just for like evaporating water to turn turbines or something along those lines. Metabolism is the sum of all chemical reactions that occur in a cell, and this is broken down into 2 main categories. You have catabolism, which is the breakdown of molecules, and anabolism, which is the building of molecules. Think like anabolic steroids. If you've got that in your head, then that should make anabolism as building make sense to you, and then catabolism just knows the opposite. We also have this concept of coupled reactions, where you're taking the products of one reaction or a byproduct or side reaction of it, and you're pairing it into another one. or you're using the heat released from this reaction to fuel the to supply the activation energy for the second one. And or for instance, there are endothermic and exothermic reactions. So you use the heat from like an exothermic reaction to fuel an endothermic, which is kind of both, because technically in that case the heat is considered either a product or a reactant in addition to supplying energy. And what this allows you to do is to carry on both reactions much more efficiently than if you just did one or the other separately. The energy currency of the cell is ATP, adenosine triphosphate. So you have your nitrogen base, adenine, and a 5-carbon sugar, ribose. You put those together and you get adenosine, and you've got 3 phosphate groups, hence triphosphate, so ATP, adenosine triphosphate. High energy compound that stores tons of chemical energy in these phosphate bonds. We use it to do chemical work for synthesizing macromolecules. We use it to fuel pumps to move things across membranes, and for mechanical energy such as contraction of muscle and cilia or flagella. A metabolic pathway is a series of linked reactions, and these reactions do not occur haphazardly. They have to be regulated specifically as to when and where they function in order for the cell to do what it needs to do, essentially. There's enough energy inside a Snickers bar to, well, essentially be a hand grenade if it was all released at once. Yet you can eat that, and because you break it down in a slow, controlled manner in multiple steps, you use that energy to fuel everything you do without burning alive, which is remarkable. Part of the way we do this is through regulating enzymes. And the enzymes are proteins that function to catalyze the speed of chemical reactions. They can speed things, but they do so by lowering the activation energy. They participate in the reaction, but they are not used up in it. And a definition here that you need to know is a ribozyme is an RNA that can act as a catalyst, which there's a theory about RNA actually being the first carrier of genetic information in early cells, because RNA is capable of being a catalytic enzyme, as well as in certain cases functioning almost like a protein and as a storage medium for genetic material, depending on how you arrange it. So it's kind of a jack-of-all-trades thing that could have fulfilled multiple roles in more simplistic early organisms. The enzyme lowers the activation energy of the reaction, but does not change the end result. So how enzymes function, this is what we're talking about here, is the induced fit model. Essentially, your substrates bind to the active site of the enzyme, forming an enzyme substrate complex, which basically just lines them up in the correct 3D configuration to allow the reaction to take place. They form a product which no longer has an affinity to the enzyme and are ejected from it, and then the enzyme is free to be used again. And enzymes can be used in both directions. You can take a product, attach it to it, and then it'll spit out the 2 reactants. Because all chemical reactions can occur in both directions. Most do occur in both directions simultaneously. It's just a question of which way the equilibrium is shifted as to which reaction is occurring with greater frequency. Each reaction has a specific enzyme, so that's one good way to regulate how quickly a particular reaction is taking place. You can produce more or less enzyme. And enzymes are usually named after their substrate with the suffix "ase." So anytime you see the "ase" on the end of something, it's probably an enzyme. So lipids, lipase, urea, urase, lactose, lactase, etc. The activity of the enzyme is influenced by the cellular environment. So there's a specific range of temperature and pH, osmotic pressure, so on and so forth, that an enzyme will function optimally in. And unsurprisingly, for the majority of the ones in your body, biologic conditions are the optimal range for that. But you can also affect the speed of the enzyme by concentration. If you don't have enough substrate, then it doesn't really matter how much enzyme you've got there if there's not enough stuff to react in the first place. So you throw in more substrate, or if you've got too much substrate, you throw in more enzymes and until you've saturated their capacity, and that will speed the reaction up. Temperature, because you're basically relying, all chemical reactions rely on the substrates just kind of randomly bouncing around until they hit each other at the right conformation. A higher temperature means they're moving around faster, which means that per second they're more likely to link up to perform the reaction. The problem is once you reach a certain temperature, you begin to denature the proteins and essentially cook them. And pH has a similar... There is an optimal range, and if you take chemistry, you'll learn about like the isoelectric point and all this sort of thing. But basically, if you have too little or too much pH, you are going to affect the three-dimensional structure of the protein and can render it inactive at either extreme. The majority of enzymes are only needed during specific times. So you may not even synthesize them. For instance, there are bacteria that are perfectly capable of producing a lactase, but if there's no lactose in the environment, they're not going to express it. But we often have a lot of enzymes that we have stored up ready to go, but we have them inactivated. And so you need a cofactor in order to bind to them in order to activate the enzyme, or sometimes the cofactor binds to it to deactivate it, and you have to have a releasing mechanism to pop well, it wouldn't be a cofactor then, but to pop the inhibitor off of the enzyme for it to function. We usually do this through phosphorylation, which takes a phosphate group and sticks it on a protein for the inactivation. Almost every enzyme is regulated by feedback inhibition, which means when the product is abundant, it binds to the enzyme's active site and blocks further production. So, like we were talking about before, you've got your substrate, your two reactants, and they will produce the end. You go through and you do your enzymatic reaction here, your induced fit model, and you end up with your end product at the end, and the end product can bind to the enzyme and inhibit it and function instead of having to phosphorylate it. So this helps maintain, and then what will end up happening here in this case is when you are low on a given thing, well, suddenly your enzymes can work again. And this kind of helps shift the equilibrium, because if the reaction wants to proceed in the opposite direction, it doesn't have an enzyme to catalyze it. So you have to go up that huge hill of activation energy, and that much energy is usually not available. So it's one way to help keep the reaction shifted in a particular direction. You have other methods of competing with or inhibiting the enzyme. You have non-competitive inhibition, where something binds to an allosteric regulatory site on the enzyme, which is similar to what we talked about up here with the feedback inhibition, but it's usually something other than the product. And typically poisons do this, cyanide in particular. But you can also regulate an enzyme with this methodology as well. And then we have competitive inhibition, where something that looks a lot like what the enzyme wants to bind to catalyze the reaction of binds to the active site and is basically wasting the enzyme's time because now it's not catalyzing the reaction it's supposed to. This is how caffeine works in your brain. We have adenosine floating around in our brain, and the longer you're awake, the more adenosine builds up, and essentially that tells you how tired you are, or it's one of the mechanisms anyway. Caffeine is a competitive inhibitor for adenosine that actually has a higher affinity for the active site on the receptor than adenosine, so caffeine comes in and drop kicks adenosine in the face and takes its spot, and then suddenly you don't feel as tired. I say suddenly, it takes about 20 minutes for it to kick in, but still. There's also uncompetitive inhibition, which is not listed here, but what that ends up doing is This right here is referred to as the enzyme substrate complex. And an uncompetitive inhibitor will bind to the enzyme substrate complex and inactivate it. Basically, the enzyme is no longer capable of releasing the substrate, and it's just stuck there. Okay, so we have oxidation and reduction reactions. The important thing here to know is oxidation is the loss of electrons, and reduction is the gain of electrons, so oil rig. And even though it's called an oxidation reaction, that doesn't necessarily mean you're sticking an oxygen to it. It just means that the thing is losing electrons. And these are usually paired into a redox reaction. So you can think about oxidation. You add your you're losing your hydrogen atoms here, so loss of electrons. Reduction, you gain hydrogen atoms, so therefore you gain electrons. So just think of it like that. Just track the electrons. Don't worry about are you adding or removing electrons. Are you adding or removing oxygen? Just track the electrons. So photosynthesis, we have chloroplasts, and they're able to convert solar energy to ATP and then use it to reduce carbon dioxide to a carbohydrate, such as glucose, or chains of glucose. You get starches and so on and so forth. And this releases oxygen as a byproduct. Plants actually also do cellular respiration because photosynthesis here just lets them store energy, essentially, but you need cellular respiration in order to use it, which is the exact opposite of photosynthesis. The difference being here is photosynthesis uses light energy to kick it off. The difference being here that photosynthesis uses light energy to kick off the reaction, and at the end of cellular respiration, we're getting chemical energy in the form of ATP that's released into the cell for performing various processes. And it just so happens that in plants, the plants end up performing photosynthesis more than they do cellular respiration, so the net is they end up producing oxygen. We use cellular respiration in our mitochondria, just like plants do for that matter, and the plants, the food that we consume is derived from plants or animals, and those animals would have then eaten plants in turn. So essentially, we are powered by plants, which are powered by the sun. So indirectly, we're solar powered, which is kind of cool to think about. So we take food, the nutrients and oxygen into the mitochondria to produce ATP, which we then use for all of our cellular processes. Which that is it for chapter 6. We'll move on to chapter 7 shortly, and if you have any questions, comments, concerns, let me know. This next exam that's coming up, I very strongly recommend that you look through the lecture summaries, because those are going to have all the information that you're going to have to memorize. Just looking through the PowerPoints here, it looks a lot less this looks a lot easier than it actually is when you start trying to answer questions about it. So look through the summary PowerPoints in order to see what it is you're going to have to actually memorize and how the questions are going to be worded on the exam.